So just a quick revision of what I talked about last time. So I introduced you to the idea of making profiles out of a collection of motifs. So a data structure like profiles. And then from profiles, in the end of the last class, I introduced you to the concept of making a profile frequency matrix, which basically tells the frequency of each of these ATGC letters at each position in the profile and then converts that into a frequency. And this frequency essentially represents the probability of finding that particular nucleotide at that particular location in the motif. And now this can be used, these frequencies can be used to predict or to basically say how likely is your current profile or the current k-mer that you're looking at in a large sequence, what's the probability of that? So for example, if this is your profile matrix, uh, and you are looking at this particular k-mer, then based on these probabilities, you can actually calculate what is the likelihood or how likely is it that you would see this particular k-mer as a part of the profile. And you can use this idea to find what's called the profile most probable k-mer, which basically is a k-mer that is the best representation of the profile that you are given and in your given set of DNA string. All right. So far, the thing that I told you about, that given set of k-mers, you can come up with a profile. And now here I'm basically telling you that given a profile and a DNA string, you can come up with a k-mer which is called the profile most probable k-mer in the sequence that you've been given. Now, this basically allows you to come up with a strategy called the greedy motif search, where you are given a profile, you take a set of motifs and from all but one DNA sequence. And then from these set of motifs, you create a profile and then you add the profile most probable k-mer. You search for the profile most probable k-mer in this DNA sequence from which you are not taking motifs randomly, and then you add this profile k-mer, probable k-mer, and then you randomly now select again set of motifs and make a profile, and then in in the DNA sequence where you have left the motif, you look for the best motif and you keep on iterating it. And this gives you a good profile. Specifically, there are two implementations of this, which I'm going to talk about. Last time I showed you something called the randomized motif search. So there are two ways of doing this, and I'll show you the randomized motif search. How does randomized motif search apply the greedy motif search algorithm? So the idea is simple. Again, you have motifs. From motifs, you can make profiles, and given a profile and a DNA string, you can come up with a profile most probable motif. So that will give you a new set of motifs from which you can make another profile and so on and so forth. And you can keep on iterating this. Okay, so this is what it shows
Sir,
here. Given motifs.
sir, could you please
Yes, Ratnesh.
briefly describe what is a profile? What is a profile?
I will not describe that because I described that in the last class, Ratnesh.
Okay, sir.
And if
Okay.
you're missing the class and you're not looking at the lectures, it's not my responsibility. I can tell you this time, but next time I will not. So a profile essentially, if you are given motifs like this, a profile is all the motifs put together, and a profile out of these motifs basically gives you the frequency of each nucleotide at a specific location in this matrix of motifs. And profile essentially is capturing the frequency of these motifs at each position in the matrix of motifs. So there is something called motif matrix. So this is representing a motif matrix where you are taking a k-mer from each DNA string. And from this motif matrix, you can make a profile, and a profile essentially captures the frequency of each nucleotide at each position in the profile matrix.
Yes, sir. Understood. Understood.
All right. So the idea of greedy motive search based algorithms is that given motives, you construct a profile, and given a profile of motives, given a DNA string, you can basically construct a new set of motives. All right. And you can keep on iterating this process until there is no change happening. All right. So this is how typically you will represent. So there are two ways of applying this set of algorithms to looking at DNA strings. The first set of algorithm is called randomized motive search. And as the name would suggest, the way that you will be selecting motives, so that's how the two algorithms differ, is how you actually select and update the motives with each iteration. So in the randomized motive search, the way you update motives in each iteration is random. So what you do is you randomly select k-mers, okay, of so if you have T sequences, you have K is the k-mer length that you're looking at, and DNA is the set of DNA strings that you are from which you are looking at the motifs. You randomly select k-mers from all the T sequences from each DNA string, right? Then you basically say that, okay, the given motif you are initializing, right? So you say that the given motif is my best motif, and now you iterate. You iterate by doing, you say that your profile is the profile of the motifs that you have got. And then based on this profile, you are going to come up with other set of motifs which will be most fitting to the profile in picture. And if the score of the motifs that you have got, and I've shown you how to calculate the motif score, basically it's the sum of all the probabilities that you'll be getting in this case. And then if the score is the least score, or the best score, sorry, then you will update the motif that you have got, and you'll keep on running this until the motif doesn't change. Okay, so it's a big question. This is so random. How would this algorithm find anything, right? And the idea here is that the inherent assumption here would be that when you are choosing k-mers randomly, you will be choosing actually random sequences, right? And if you actually random sequences are picked, then the probability of each nucleotide at each position is going to be roughly 1/4 because it's absolutely random, right? But typically, you know that your sequence contains a motif. So that essentially would mean there is some order there in the k-mers that you will be selecting. And because you have some order of k-mers, the profile is going to point towards the frequencies are going to point towards the real motif. And we are going to see that how that works. Okay. So let us say you have these 5 DNA strings and we have implanted a 4,1 motif of ACGT. So this is ACGT. So this is ACCT, ATT, ATGT, GCGT, right? So on and so forth in these 5 sequences. All right. So now what you do is, let's say you randomly pick up 4 mers, right? So K is equal to 4 here in these 5 sequences. So these are the motifs that you have randomly picked up. Here we have picked up TAAC. Here we have picked up GTCT. Here we have picked up this one, CCGC, ACTA, and this last one. Okay, just by chance, sometimes you might pick up a motif that represents your original motif, right? Your original k-mer that's a part of the motif. That may happen, right? Just by chance. And sometimes these are overlapping, and sometimes these are absolutely not overlapping, right? So now what do we do? Out of these motifs, we construct a profile. So these are the motifs, you line them up. There are 5 sequences, there are 5 k-mers, and out of these 5 k-mers, you create a profile. So can somebody tell me what is going to be the probability of finding a T in the first column?
Point two, point two.
Be 0.2, right? So T is 1 by 5, G is 1 by 5, so on and so forth, right? So you create a profile for these particular motifs. And now what you do is you take this profile and you go back into the sequences, into the strings that you've got, you extract k-mers out of each of these profiles and you score them, right? You score them to find the profile most probable k-mer. Now, when you do that, when you go back, you find, okay, this profile hits at the right k-mer, this one, ACCT. The second profile also hits the right k-mer. The third profile, the third string also hits the right one, so on and so forth. So funnily enough, in just one iteration, because of random search, and this is truly random, right? This is at the end. This one is overlapping a little bit. This one is also not overlapping at the end. This is the beginning. This one is also not overlapping at all with your sequence, and this one is totally overlapping, right? So this is something that could truly randomly you could come up with. And now actually when you do this only once, you hit the motifs, right? So the idea here is that when you select k-mers randomly, it results in a uniform expected profile where every entry is 1/4. Okay. So such uniform profile is useless because it does not provide any clues about the implanted motif. But that is not the case. DNA strings are not truly random. They already include implanted motifs. All right. So that essentially means that the profile that you're making, even with this random selection, would be biased. So the implanted motifs result in biased expected profiles. And these profiles are always pointing towards the location of the true motifs, right? Because that's how you are updating. Your profile is giving you motifs that are most like the profile, right? And then when you make the new profile is again going to point to motifs which are more like it, right? More like those motifs. So essentially, you are gravitating towards those motifs that have more similarity inherently. Okay. So again, showing you another example here, right? Or, sorry, not like another the same thing. So this one, because you had the correct motif, one of the correct motifs, right, of your profile. So this correct motif sort of points towards the right direction, right? Because you have a here. So if it was all random, it would be same probabilities. But because you have one of the correct motifs here, so you catch the correct probability here. Again, the here the probability is sort of updated in the same ways, right? So statistical bias points to the direction of the implanted motif because one of the randomly selected formats happens to be the implanted pattern simply by chance, right? Or some of them are overlapping the implanted pattern by chance. Is this clear to everyone?
Yes, sir.
Sometimes coming up with random algorithms or coming up with random ways of solving the problem might not be the worst situation, right? And this randomized motive search currently is if you can see, it's much faster computationally than what we have discussed so far, even much faster than the median string problem that we solved in the last class. Okay. Now, why is this important? And can we implement this again to a biological problem? So last class I discussed about circadian rhythm. Today I'm going to discuss about another biological problem, which is talking about a different set of genes, for example, that where these kind of motifs might be coming in. So the question is, how do bacteria hibernate? So has anybody gotten a tube test for tuberculosis here? Any one of you? Any one of you has gone for in an internship abroad? Okay, so if you haven't, then this is something that most of you who would be either going for a job or an offsite or an internship, this is a test that everybody will get done. So this is called the Mantox test. And this basically what it does is it injects a very purified form of a glycoprotein which is found in tuberculosis, which is found in tuberculosis cultures. And people who have tuberculosis already or have been exposed to the bacteria, they basically they elicit immune response against this protein. So whenever you have this small protein injected under the skin, after you see that after a few days, if the person is, depending upon the category of exposure that this bacteria has, you would essentially see a larger kind of immune response on your skin. So this Montauk test is something that if you haven't undergone, specifically in your US and Europe, whenever you'll be going and spending long time there, they would ask you to do this test. So why is it important? This is important because you are detecting tuberculosis, and tuberculosis is a big problem. About 2.2 million cases of tuberculosis are happening in India. It's about 22 lakh cases every year. 26% of all the world's cases, and specifically 38% of the deaths out of tuberculosis of the world's cases are happening in India. So India is having a very high tuberculosis burden. That therefore most Indians, whenever they are going abroad for a long period of time, they are tested for tuberculosis. Um, And because tuberculosis has been there with us since the 19th century, so it was basically a big tuberculosis outbreak happened in Europe in the 19th century, where I think 1/4 of the people got it in Europe. And because they were invading us, so I think they brought us the tuberculosis as a gift. Um, because tuberculosis has been there for such a long time, so over 200 years, we see that there are a lot of tuberculosis strains which are resistant to a lot of antibiotics, and therefore it's just very hard to get rid of uh, this disease. Now, given this, actually 1/3 of the world's population, it's so hard to get rid of it. 1/3 of the world's population actually carries latent tuberculosis in them. Now, what do you mean by latent tuberculosis? Latent essentially means that you have the bacteria residing in the body, but not reproducing so fast, right? So it's like flying under the radar. So the question is, how does tuberculosis, the MTB, know to switch from the latent state to the active state? Because when it switches from the latent state, when it's not reproducing, to the active state, when it is reproducing, when it reproduces, it causes disease. All right. So this is very important to know, because when it does so, tuberculosis happens and people die. And this is something that we have to worry about. So this hibernating form of tuberculosis typically forms metabolically dormant spores, and they can survive very tough conditions. Now, whenever they are forming these metabolically inactive spores, they essentially are relying on very little resources. And typically, very little resources also is associated with lack of oxygen. So, for example, these latent forms of TB can survive in hypoxic conditions. Hypoxic conditions essentially mean that you have very little oxygen. So just like biologists found that there were three genes controlling all the circadian rhythm genes in plants, 1,000+ circadian rhythm genes in plants, they found that 25 genes were associated, the expression of those genes, essentially meaning that these genes were expressed, they made protein, when you had significant hypoxia given to these tuberculosis bacteria. So now the reverse question is that you have these 25 genes, and can you actually tell why these 25 genes are coordinatedly getting expressed when you are giving hypoxia? So the question is that the expression of this gene might be controlled by a transcription factor, and they call this transcription factor creatively the dormancy survival regulator. And they want to basically find out what are the motifs these dormancy survival regulator is most likely binding to in these genes and therefore regulating them. Okay. So now when we apply the two algorithms that we have discussed, the median string search and the randomized motif search, to discover the regulatory motifs that control the expression of these genes and might be the locations where the dormancy survival regulator, this protein, might actually be binding, we find something interesting. So we have no idea of the length of the motif, right? Because we are starting from scratch. If we apply the median string search algorithm, these are the results that we get. And if we apply the randomized motif search algorithm, we get slightly different results. And for randomized motif search, actually, you can go all the way to k equal to 20, the size of the motif that they can find. Can somebody tell me that why in the median string search, we have restricted ourselves to k equal to 12 and haven't gone beyond that?
So perhaps the runtime will be very high, or maybe our system will not be able to
So why is that
calculate.
Why is that dependent on k?
Sir, like factorial of k is equal to 2, we cannot something like calculate with using the Python. I guess something like that. There may be.
So does anybody remember the runtime of median string?
N to the power t
4
into
to the
k into t.
power k.
So n to the power t was
Okay,
for
sorry.
the
Yeah,
other one.
that was
So 4 to the power
for
k,
the
right?
yeah that yeah.
Yeah.
Yes,
So
sir.
Ashi said 4 to the power k, right? Yeah. So 4 to the power k, right? So that essentially means that by having from 4 to the power 12, if you go to 4 to the power 13, it becomes 4 times more complicated, right? And 4 times more computation has to be done. So typically these median search algorithms do not go beyond 12, 13. But randomized search, because these are much faster, you can actually go all the way up to 20, right? So that's one thing. The other thing is that you have these different kinds of motifs come up. So one set of motifs that come up for a median string and for your randomized motif search, they are, let's say, the smallest one. I mean, they are sort of different motifs, right? That they are coming up, that these algorithms are finding. So that essentially gives you two observations. The first observation is that these algorithms are not always giving you an exact representation, right? The exact motif, right? So these motifs might be uh, sort of different algorithms might give you different motifs, right? So now there has to be then one way of finding out. And the other thing is that the motifs that these different algorithms might be giving you might actually be of different lengths. So how does one assure ourselves that the motive that we have got is the correct motive? Are there any ideas? There is some idea for it. If you basically look at this area, can you look at this area and tell me like which motive is probably the best motive between median search and randomized motive search? Can't be that hard, no?
So isn't it that based will be based on the probability of all these base pairs, and then we will be finding that.
Uh, we're going to come to that, but can you look at this slide and tell me? So how do we actually find the motives from the algorithms? What are we optimizing for when we say that, okay, we have this motive, this is the best motive? What do we calculate?
Score.
We
For the
calculate a
score.
score, right? So basically there is one scheme of scoring motives, right? It is coming up with a score. So you can compare scores of motifs of the same length, right? So if you compare the scores of k equal to 12 here between this motif and this motif, then you see that the motif with a better score, in this case, depends how you are scoring if you are calculating the number of mismatches. So in this case, the one with a lower score is going to be the better motif, right? If you were to do with the greedy strategy, then the one with the higher score is the better motif, right? So it's based on a score. But what happens when you have motifs of different lengths? Can you compare their scores? Right? So it's not straightforward to answer this question. So we have to come up with another way of actually defining whether the motif that you have got, how much information is being captured in that motif in the context of the k-mers that represent it. And I'll explain what do I mean by that. So by information, I mean, uh, so basically by talking about the lowest scoring one, and by telling which motive captures the most information of the k-mers informed and underlying, then we can essentially say that this is most likely the real binding site that we are looking at. Okay, so let us say a specific motif is represented by a certain profile. All right, so profile as given here. Now every column of the profile essentially is a probability distribution. So right, there's a probability distribution. So A, C, G, and T, there's a probability distribution for each column. Now the probability distribution of each column actually describes the entropy of each column. So we define a term called entropy, which is another way of saying that is another term for the probability distribution itself, right? And sorry, so the entropy of a probability distribution can be calculated as minus P of that specific column multiplied by log 2 of P and sum of all probabilities. All right. So let's see if we do that, what kind of values do we get? If you have a uniform distribution, right? That means A, T, G, C are all equally likely. What would that indicate? Does that indicate that that location, that particular base is important in the K-mer or not important in the K-mer?
Not important.
Not important, right? It's uniformly distributed. So if a protein, let us say, is going to bind using this motif and any of the four bases can come at that location, then that protein is not specifically deciding whether it has to bind to that DNA sequence based on that location. So that location is not informative. Does everybody get this?
Yes,
Off
sir.
hands, please. This is very important topic. Okay. Only three people are listening or the three people have got it.
Sir, one question,
Okay.
like why here entropy is defined as negative?
It's just a mathematical term that we have here. It's not defined as negative. It's actually minus of these probabilities. And because the probability is between 0 and 1, right? So if you take the log of something which is 0 and 1, it will be minus, right? And so
Yes.
this is just a, yeah, you're doing minus sum of all the probabilities here. Okay. Okay, so only a few of you have gotten it. So I am still going to go ahead because I don't see many other people participating. So the idea here is that you define a number which tells you how non-random a column is or how non-random the sets of columns are when you are summing all these probabilities together. Okay, so at each of the column, if everything is absolutely random, then it has maximal entropy. That is, there is maximum. That's what entropy is like. Entropy is like randomness, right? So there is maximum entropy. So this value is 2. So this is the, if you get a value of 2, then that essentially means that that position is contributing no information to that specific position in the profile. Similarly, if you have a completely conserved column, so their probability is 1, that you have only say one, only a G being found at that position in the profile, and all the other probabilities are 0, then essentially the probable this value becomes 0 if you calculate it. So log 2 of 1 multiplied by 1, right? And 0 multiplied by all the other probabilities, that will be 0. So this probability basically becomes 0. Okay. So why am I telling you all this? This is one way. Now, if you would to sum all these values together, okay, all the entropy values from 0 to 2 of the first column, you will get a singular entropy value, okay, for that particular column. Like this, you can get entropy values for all the columns, right? So that essentially tells you that by adding one more column or by having a k-mer which is say from 12 to 20, by adding 8 nucleotides, are you actually adding more information or not? Because all this information has been scaled for the amount of new information. So this score has been scaled for the length of the k-mer that you're having, right? Length of the k-mer that you're. So basically, this information would essentially tell you what is the average entropy that you're capturing per motif. So the entropy of the motif is the sum of the entropies of the columns of its profile matrix. All right. So now when you would be seeing a profile like shown you earlier, so this is how this profile is typically represented. So 12 mer is going to be represented like this. So the height of the each nucleotide will go to 2 because 2 is it's basically the height is going to be 2 minus the entropy, sum of entropy, right? The total height of 4 nucleotides is 2 minus sum of the entropy that you have at that position. And then the relative entropies of each of the bases is going to be representing the height of each of the bases with respect to the entropy that is there at that position. Now what you can do is you can compare the entropy of the total motif between two different k-mers, and you can say whether the k-mer that you are having is actually capturing more information or less information than a random motif would do, than by adding random bits or random nucleotides to the two motifs. Is this clear to everyone? So if you ever see a motif logo like this, now you know how to interpret this information. Yeah. So if I would give you a profile
Yes,
motif,
yeah.
can you read? Can you make a motif logo like this? And also, given a motif logo like this, you can interpret the profile motif.
Oh, yes, sir.
Yes, sir.
Yeah. And you also know how to now differentiate, basically say that a motif is the best motif, right? Based on scoring or on entropy based scoring that you'll be performing. All right.
Yes, sir.
So the predicted versus the real motif. So this is the randomized search motif, right? And this is motif of the real binding sites that you get. And you see that there are a lot of similarities. So there are a lot of G's that were predicted. And you see that in the real motif also there are a lot of G's. You have a C here and you have a GC here. So these things are also kind of very similar. So if you actually read and compare this, so this motif reads as AGGG. At here, you can say I think C and A are similar. So this, let's say it's an A here. Then there is a C, G. There is A, then again A, then a G, then a T, C, C, C. Then you have T, and then you have A. So basically the letter which has got the larger probability, the larger entropy, is basically at or smaller entropy is smaller contribution to entropy is kept at the top in such motifs. And whenever you are given such a motif, you have to read from the top essentially to find out the motif, right? So this randomized motif search gives you a very good idea. Basically, what I'm trying to show you is gives you a very good idea of exactly what the real motif actually was. So can we actually speed up the process even faster? So I showed you the randomized motif search. Can we actually do this even better? So to do this even better comes the second way of implementing your greedy motif size algorithm, which is through Gibbs sampling. And as I told you earlier, the way that you differ between the two methods is how you are updating the k-mers that you are selecting at each iteration. So in the case of randomized motif search, you start with a random set of k-mers. And then after starting with a random set of k-mers, you are finding the profile most probable k-mers for each of the DNA strings which are there in your input. All right. So there is a possibility that there is a possibility that the original k-mers that you had selected, right, randomly, it might have one or the other correct k-mers, right? But because each time you are trying to update and find all possible profile most probable k-mers, it's possible that you might just discard all the k-mers that you initially started with. Okay. So this essentially leads to the problem that if you are starting with very large sequences, very large DNA strings, then you will be seeing many more k-mers, right? So if you're seeing many more k-mers, and just because you are randomly doing this process, you might actually be comparing, you might be statistically biasing your profiles towards more randomness. How does Gibbs sampling overcome this? Gibbs sampling basically overcomes this by not updating or not giving the algorithm a chance to update all the k-mers, but at each step you will only update a single k-mer. Okay, how does that differ from the randomized motif search? In the case of randomized motif search, at the end of the iteration, you might just quickly and randomly, not randomly, you might quickly replace all the motifs that you started with. So therefore, you are not giving that much weight to the profile that you started with. Okay. On the other hand, when you are doing Gibbs sampling, in Gibbs sampling, at each point of time, you are only going to update a single k-mer in each iteration. So you are moving more cautiously, and you are saying that, okay, the profile that I must have started with might not be that correct, right? But so I'm just going to slowly move in the space of k-mers that I'm getting. And I'll show you how that helps and how that actually makes things better. So to demonstrate Gibbs sampling, we start to take slightly longer k-mers and we start to take slightly longer sequences. So earlier I was showing you a k-mer of length 3. I'm going to show you a k-mer of length 4 now. So here are these 5 sequences. And let us say we have taken these k-mers, right? Again, we have selected these k-mers. So TAAC, that's actually the same example. TAAC, you have these, this is the bold one, GTCT, this one, and this one. All right. So what we do is now we will randomly select one of the k-mers and that DNA string, and we will remove it from the motif profile that we are going to make. So we randomly remove this third line, let's say. Okay. And we will basically be updating the k-mer in only this third line. That's how we do this Gibbs sampling. So when we randomly remove one of the lines, then the profile that we'll be having will be based on 4 sequences only. Okay, 4 k-mers. You'll be making the profile, you'll be making the frequency matrix, and then you would essentially calculate the probabilities of all the k-mers that are going to come based on the profile matrix. All right. But now instead of updating things based on the score of the gamer, you are going to update based on the probability that you are getting. So essentially there are two probabilities that you are getting, this 1 by 128 for this motif and 1 by 256 for this particular motif. So now you are essentially going to toss a loaded die, right? A loaded dice. And you're going to say, okay, what is if there is 2 times there is probability of getting this and there is 1 time of probability of getting this particular gamer. I'm going to roll the dice and I'm going to see, okay, do I get this or do I get this? If I get this particular gamer, then I'm going to say, okay, this is the gamer that I've selected this time. And I'm going to now update my matrix. And I'm again going to repeat this step. Okay. Again, I'm going to repeat this step. I'm going to say, okay, this was my new gamer. So in this case, they rolled the dice and the gamer that they found was GCGT. And then they say that this is the updated one. And now they will again repeat this process by randomly selecting one of the lines. So let us say they will select the second line, right? And they will update the second line. We will update the second line in this algorithm, and we'll keep on repeating it until there are like no changes for over say 100 iterations. All right. Now this, however, leads us to a problem, and that is the problem of zero probabilities. So the problem of zero probabilities essentially leads us. You cannot essentially have a dice which has zero probabilities for something like this, as I will show you in the coming slides. Okay. So you found that this was the new gamer, and now you will iterate. So this is the Gibbs sampler outline. You form motifs by randomly selecting k-mers in each sequence. You randomly choose one selected k-mer from the removed sequence and remove it from the motifs. Okay. You create a profile of the remaining k-mers. For each k-mers in the removed sequence, calculate the probability of the k-mer for that particular profile, resulting in n minus k plus 1 probabilities, because this is the number of k-mers that you'll be having. Now you roll a dice which has these many sides based on these probabilities, and basically whatever with that particular loaded die that you have, whatever probability comes, whatever combination of the gamer comes, that gamer becomes the new starting position of the rolling die. You add it to the to your original matrix, and then you repeat steps 2 to 6. All right. So here, instead of iterating based on the greedy algorithm, always choosing the right or always choosing the best k-mer, profile most probable k-mer, you assign probabilities to k-mers and then you select based on the relative probabilities that you have that you will be getting. So, but that would mean that you will never, in this kind of approach, you are never going to get to you're never going to sample these other k-mers, right, which have zero probabilities. Now that is a problem, and we'll see why that problem is a problem, right? So you have these zero probabilities as shown here. So because if you are not going to hit or not going to like update these k-mers, they might actually be having the real motifs. They might be representing the real k-mers that are part of the motif. And because of this kind of a issue uh, with zero probabilities, you might not be able to update them. So we have to come up with a way to solve this problem. We cannot roll a two-sided dice. So here I introduced you to something called the Cromwell's rule, which essentially is another way of saying never say never. All right. So Cromwell rules say that we should avoid the use of probabilities 0 and 1. Right. So why am I saying that? So I'm saying that because the k-mers that we are getting as a part of our algorithm, we do want to give them some finite chance to be sampled, right? If they do not contribute to the probabilities, if they are not contributing, even then you want to give them some finite chance because there is a possibility in this random sampling that they might be belonging to the original motif, right? If their probability is not good enough, that's fine, but there should be some finite probability. Okay. And this is what Cromwell said. And this is what we will try to figure out by introduction of something called pseudo counts. So pseudo counts is a problem, and that has been solved. Sort of a solution for that has been given by Laplace. So basically, in small data sets, there is always a chance to observe event to basically say that a specific event might not be might not occur because you haven't seen it enough number of times. Okay. So specifically in randomized algorithms, you do not want to have this kind of a problem because it randomized algorithms rely on your ability to at least sample the positions. If you have zero probabilities, then you will not be able to sample these positions, right? So to give you an analogy for this, if you were to find out the probability of having the sun rising tomorrow, right? Uh, and you were to observe for say 5,000 years that sun rises, right, every day, and sun rises the next day. But you haven't observed so far at least one day that the sun hasn't risen the next day. That possibility is still there because you haven't observed it. So that's what Laplace said, that unless you have observed something, the probability of observing that event is still one out of all the observations that you have made. Okay, so this is basically called adding a pseudo count to your probability matrices. Okay, so how do we do this? So basically the formal line for this is, if we repeat an experiment n times and you have a success or a failure n times and you get like s successes, the probability of finding the next repetition will be a success is not s by n, but it is s divided by n plus 1 because you haven't seen the failure yet. Okay, so just to show you the mathematics are behind it. So if there are x1 to xn plus 1 conditionally independent random Boolean variables, failure or success 0 and 1, and we talk about Boolean variables because your nucleotides are also Boolean in nature. They are not Boolean, so they have like 4 options. So they are like a discrete variable with 4 options. So then the probability of x n plus 1 is equal to n given that x1 to xn, so the past n events, they were a success, is s by n, right? Since we have the prior knowledge that both the success and failure are possible, all right? So we have already had two observations, one observation of success and one observation of failure. So the n becomes n plus 2, all right? So therefore, we are adding these pseudo counts which tell us that we are able to define success and failure as well in our experiments. And the probability that we are going to see a success is also going to be updated because you know that the success can be seen, right? So is going to be s plus 1 divided by n plus 2. Oh, how does that affect our
So
matrices
I had
that
a
we are
question here.
constructing? Yeah.
Uh, so for example, if we take the example of a coin toss and like say if you get a heads, you win, and you toss a coin 10 times and you get 10 heads.
Yes.
So, but the probability of getting a tails on the next toss is still half, right? It's not like 10 by 10 or 10 by 11.
So I mean, in this case, it will be half if you are assuming that the coin toss is not loaded. Only
Yes, sir.
then you can say it's half. And
Yes,
you
sir.
say it's half because somebody has observed in past that when you toss the coin enough number of times, you see equal number of heads and tails, right?
Yes, sir. If
But
we are getting
when you
like
are
10 out of
Yes,
basing,
sir.
but when you are
10 out of
basing your
10,
observation up, please let me finish, Ratnesh.
yes, sir.
One, if you are basing your observation, so you are saying that I tossed the coin 10 times. In my experience, the probability is 10 by 10, right? This is what Laplace is talking about. He's talking about an experience based probability that you are telling because you don't know, right? Here you are starting with no information, no prior information when you are working with these kinds of algorithms. So you are basically basing your judgment based on the observation that you have, and the fact that you can observe a success and a failure is already two observations made. That's what he said. So in this case,
Yes, sir.
the right way of saying the correct probability would be that I know that there can be a heads in my coin, right? You can take the coin and you can put it on using your hand and the tails will come, and you can put it using your hand and heads will come, right? That's how you know a heads and a tails are possible. So you've already made those two observations. Now you toss your coin and 10 times out of 10, the remaining 10 that you have done, you have a tails, right? So now the probability is going to be 10 times divided by 12. But according to Laplace, it should be 10 times plus 1 divided by 12. So it should be 11 by 12 instead of 10 by 12 or 10 by
Understood, sir.
10.
Yeah,
Make
understood.
sense or no?
Yeah, yeah.
Sir,
Yeah. Wait, I think
sir,
Ratnesh has
can
a
we also
question.
say,
Ratnesh, maybe if Ratnesh asks first, then you go. Ratnesh, please.
No, sir, nothing, sir. Okay.
Understood?
sir, can we
Yes,
also
sir.
say that in a greedy algorithm, since we do not consider all the data and the algorithm might uh, not be aware of the future choices it would be making, so that's why we go with the precaution of adding one.
Huh, exactly. I mean, the point is that future choice, we don't know that it will be making the future choice. That
Hmm.
is one thing. And the other thing is also that we want to give at least some finite chance, right? To actually
Right, yeah,
scan that
exactly.
k-mer.
Mhm.
Agar that chance for all the other k-mers, given the profile that you have is zero, so
Zero,
there'll
right.
be never you will be updating, never you will be going to that k-mer, right?
Mhm.
So that's
Okay.
the whole idea that it should be for it to be truly random, you should be able to visit each of the k-mer at least once. Okay, Satyakam?
Okay, sir, yeah.
Yeah, and I'll show you how that works now. So let us say we
Mhm.
have the same thing earlier, right? Same thing, we have the same five sequences, we have the same k-mers selected. We delete the third one, we make the profile, and now we have the count matrix like this. Okay. We say, okay, the ACGT, it's 2011, right? But now we know that A can come, C can come, G can come, and T can come, right? All of the four nucleotides are possible. So what you do is you add a 1 everywhere. Okay. So something that was random here, for example, still remains random, right? Something that was 0, however, now that changes. You give it some finite probability. It's small probability. It's scaled with respect to all the other probabilities, but there is a finite probability now. So you update your matrix like this. And now when you do this, the probabilities that you will be having are going to be updated as well. The two k-mers which were coming are still going to be having more better probabilities than the other k-mers, right? So when you will be tossing a die to select which k-mer you should update, still these ones will be biased, but the other small ones are still going to have some finite probabilities to get updated. And this allows you to explore the space of k-mers better in a Gibbs sampler. Okay. And when you do this, you are then able to properly sample the entire space of k-mers that will be generated from each DNA string. And as a result of which, you will be able to slowly but surely move towards the answer. Okay. So let us say we did this, we updated, we said that this is the one now. Now let us continue. Let us say randomly the first one was gone. Let's make the k-mer matrix, counts, updated pseudo counts, probabilities. You have calculated the probability of k-mers, right? These are the probabilities. Let us say this is the most probable one. Let us say, oh, sorry, not the this one is the most probable one. AACCT, right? You roll the seven-sided dice. Let us say you land on AACCT. It's most likely you'll land there, right? And then you iterate. Okay. You get this one. I think this one is wrong, but yeah, by doing this kind of thing, you will get this is the most probable one, and then you are basically within 2-3 iterations, you have gotten this, but basically by doing this, you're at each iteration, you're replacing only a single k-mer. You're not replacing all the k-mers. Okay, is this clear?
Yes, sir.
So whenever you are doing this kind of Gibbs, like a random motif search, it greatly enhances the possibility of you uh, looking at sweeping a large sequence space, a space of DNA strings. And by introducing a Gibbs sampler in the random motif search, you can do it more slowly and surely towards the pattern that might be implanted. But now the problem is that if you talk about patterns themselves, like these regulatory motifs, it might be a big problem if you have even the best algorithms will fail if you are looking for patterns which have lots of mutations in them. So for example, a typical motif that we talked about, that motif that works for tuberculosis dormancy genes, is basically a motif. It's like a 15,5 motif, because if you see these 1, 2, 3, 4, 5, 6, 7, 8, 9, uh, 10, right? These about 10 bits are having a lot of information, so they would usually not be changing. And 1, 2, 3, 4, 5, these locations are not really good because they seem to have lot of lot of other possibilities of nucleotides. So even if you are actually looking for a 15,5 motif in which is inserted in about 1,000 nucleotides long random sequence, most of these motif finding algorithms fail, right? So that doesn't mean that you sort of give up. Uh, but what happens is that biologists have found a way to reduce this search space. So instead of looking at 100 nucleotide long motifs, biologists have come up with a method called chip sequencing, which is called chromato-immunoprecipitation followed by sequencing. It's a short form for that. This is essentially an experimental technique where you can precipitate, you can using an antibody against the protein of interest, you can precipitate that protein and the DNA which is bound to that protein. And typically you will be getting approximately 100 to 200 nucleotide long DNA that will be bound to the protein of interest. And then if you are able to sequence this DNA that is bound, the DNA which is bound to the protein of interest, and you know the identity of that DNA, once you know this, then in that small fragments of DNA, you can actually look for motifs and can say whether those DNA fragments are enriched for a specific set of motifs that might be predicting uh, the binding of that protein. Okay, is this clear? So I think I'll stop the class today. I'll wait for questions. Otherwise, you are free to leave for next class. So I highly recommend all of you to please uh, do some of these exercises yourself of sampling and random motive search so that you get a hold of how these things work. Yeah, because there can be some twists that can be added to the algorithms to allow them to solve much more complicated problems or a different problem. But the ideas remain the same of how you would be randomly selecting things, constructing profiles, and scoring, or you would be doing this and then replacing the k-mers based on a greedy strategy, totally random strategy, or you can be replacing them based on a Gibbs sampling strategy. And if you have motifs, two different sets of motifs of different lengths, how do you actually calculate the entropy? How would you compare the entropy? These are the kinds of things that I want you to take away from it. And in tomorrow's class, in the lab, I'm going to introduce you to one tool which is called meme, actually MEME, which actually allows you to, which is a tool that searches for, that implements these motif finding algorithms for a very large set of DNA strings. And if they are used very widely in bioinformatics. Are there any questions? Okay, if there are no questions,
Sir, I
see.
have a question.
Yeah.
So the thing is that like, uh, uh, how do we know that like bacterial chromosomes have one OIC? Like, is it the case with most of the bacterial chromosome? Uh, Definitely, it should not be like with all the bacterial chromosomes. Sir, like, I just wanted to know that, like,
Okay, so maybe
do we
Ratnesh,
have
you can
papers
write
like
this.
some?
You can write this question to me in Teams so that everybody else
Yes,
can
sir.
also know the answer. I will try
Yes,
to
sir.
find the most consistent answer which is out there and I will give you the answer. Okay.
Yes, sir. If some paper will be available, that will be better. Like I see that
Okay, I'll do
like
that.
on
I'll do that. I'll
vector
do that. I'll do that. I'll do that. I'll do that.
there are plasmids like 4 to 5 kb plasmids in which we construct two OIC